most important is, how many of you are teaching middle school? Okay, and then the rest high school? Yes? Okay. All right, and then how many of you are biology? Okay, chemistry, math, computer science? Okay. Anybody I left out? Earth science, thank you. Okay. So I'll try to tweak it as much as I can to make it as applicable to everybody as possible. All right. So, let's see here. How many of you have heard of bioinformatics? Raise your hand. Okay. All right, anybody teaching it or trying to teach it a little bit? Okay. All right. So, my goal for today is to try and make bioinformatics not scary, because it was really scary to me. Because I was like every other teenage girl, I wanted to be a marine biologist, right? That's what I always wanted to do, and I did that for a long time, and I still get to do a little bit of that. I wanted to study sharks, so I wasn't quite like the dolphins. That's the right? That's the typical. I wanted to study dolphins, right? So I really wanted to study sharks because they're what we call charismatic macrofauna, right? Nobody wants to study things they can't see. They want to study sexy things that are big, that right, are on television. Yes. So that was what I wanted to do too, and I didn't want to do this at all. And now I love it. And so I want to try and help see how you can get students from point A to point B. So my story. So I went to Virginia Tech for undergraduate biology, the Hokies, and I went to Clemson for graduate school. So my Ph.D. is actually in zoology. And my PhD advisor wanted nothing to do with molecular biology. He thought that people who studied molecular biology were not true scientists. They didn't understand the history of science. He was really old school. And so I didn't get that training initially. I was really doing kind of organismal type biology. And then when I went on to my postdoc at University of Maryland Medical School in the Department of Microbiology and Immunology, I started to learn some of those techniques and was able to keep doing that as I went on. So it's really been kind of a process to get to what I'm studying now. So I want to tell you a little bit about bioinformatics and teaching it in the classroom. I've never given this talk before, so we'll see how it goes. All right. So what is bioinformatics? And I usually like to start by telling you why you should care, right? Because otherwise your students aren't listening to you, right? And so I'll just give you a quick peek. There are jobs here. Jobs for your students, okay? So I'm going to talk about that in just a second. All right, so this is why you care. So bioinformatics. So using computational, mathematical, and statistical approaches to probe and analyze massive sets of biological data. That's the big thing. And you've heard of big data science, right? It's all over the media. Okay. And it involves developing and utilizing computer databases and algorithms to accelerate and enhance biological research. So we're talking about the intersection of biology, chemistry, math, computer science, all together. And it's more of a tool. It's not really a discipline per se. It's like a technique, yes? So you could be asking questions about lots of different things, but this is a tool that you could be using today. All right, so let's get oriented here. Right, because when we're talking with students, we have to start with what they understand, right? So we're starting with our person, right? And then we're going to zoom in on, I don't know if my pointer will work well. Then we're going to zoom in on our cell. And then inside our cell, we're going into the nucleus. And then most students know just from basic biology that they've had in middle school. My 7th grader knows that DNA is in the nucleus. Okay. So we're all right. And often they'll recognize chromosomes. I'll ask them, show me what your chromosomes look like, and they all go. Okay, so they've got their chromosomes here. And then we're zooming in on that chromosome, right, so that they can see how it's all bound together and the proteins that hold it together. And then we zoom in even more. And most of them know DNA is a double helix. So we're like, okay, I'm good. And then we take the last step where they might not be good, which is right here, where we're talking about these A's and G's and T's and C's, the bases that build the DNA. Yes? Okay, so everybody can understand that. All right, double watch right here. Okay, let's zoom in a little bit more, think about this a little bit more. So here we have our DNA, and DNA gets transcribed into RNA, and then gets translated into peptides. Peptides, we put a bunch of peptides together to make polypeptides, and those polypeptides make the protein. And proteins are what make us work. Okay? Alright, so we're good. Alright. So, with that background, I want to talk for just a second about omics, because omics are all over TV and Dr. Oz, and you'll hear about omics of all different kinds. And if you're really getting into biology and science more, you're going to hear about all different kinds. So I want to introduce this. So the most simple is genomics, so studying genomes. And then we also have proteomics. So what's proteomics? Right, we're studying proteins. All right, transcriptomics. So we're studying transcripts. So we're going from the DNA to the RNA. So we're really studying the RNA. I'm doing this one and this one. I'm not doing proteomics right now. How about metabolomics? So now we're studying what proteins or genes are involved in metabolism only. We're narrowing the field a little bit. All right. There's every omic you can think of now. Like all the scientists are coming up with their own. So now there's the inflammasome and the immunosome, and they're just making up words. Which is fine, because we know what it means, right? We get it, right? We're studying the genes and the proteins that are involved in these specific functions. And it's that fun today. The moment at the end. Okay, this is where that field has been going. All right, let's talk about timeline, because this is all new stuff. So what's been the timeline for this? So DNA was first sequenced in 1968. In 1977, the first virus was sequenced, so we're starting small. All right. And then the first bacterium, we're getting bigger. And then we made it up to a yeast, so we're at a eukaryote, more complicated. And then first genome sequence for a multicellular eukaryote. So C. elegans is one of the model species in biology. It's a nematode. And then 2001, the first draft of the human genome was released. So that's our timeline. Pictures for all of them. All right. Okay, so with that in mind, that timeline in mind, I want to tell you, what does that mean? I said sequence. What does that mean? So guess. What does it mean? Put it in order. What are we putting in order? Not just the genes, but those A's and T's and G's and C's that make up the genes. That's the order that we want to find out. So let me tell you a little bit about how that happens. But in the snapshot one slide, this is it. This is how you do this technique. Okay? So we start off with our B, we grind our B, and extract DNA. And you can do this in high school biology labs all the time. Okay. And then they break the DNA into pieces, and there are different ways that gets done depending on the technique they're using. And then they're going to sequence the pieces. Okay, I would need a whole nother lecture to be able to talk about the chemistry behind how we sequence, and that's not why you're here. Okay, so we send out our samples for sequencing, and they send us back a giant pile of data that I then have to dig through to find the parts that I need. And it's like a puzzle. So that's what I wanted to show you here. So here we have our pieces, right? And they sequence the pieces, and here you can see that we got the letters back for each piece. Yes? There it is. We can see we got the letters back. We don't know what they are yet, or where they go, or any of that. But then we can use these computer programs that will match up the overlapping parts and piece it all back together. Do you see how that works? You just match the ends and piece it all back together. And we don't just do this once. The genomes that I published this year, we have either 50x or 100x coverage, which means we've checked that join right there 50 or 100 times. We sequenced it over and over and over and over and over and over and over. All right? So we can be pretty confident. In the literature, if I have 50x coverage, nobody's going to doubt that my data is reasonable. Okay? 100x coverage is kind of overkill. All right. So that's how we do it. So I can't teach you about the sequencing and I can't teach you about the computer programs and the nitty-gritty of those, but those are great topics to try and dig into with high school students for sure. There are things that high school students can do there. Okay, so we've ended up with our whole sequence down here at the bottom. Okay, so that started out with like the most basic, what we call Sanger sequencing, like the original techniques for sequencing. And so now we've moved on to what's called next-gen sequencing. And next-gen sequencing has really revolutionized this field. And that's why, if next-gen sequencing hadn't happened, I wouldn't be talking to you about this. Okay? So, it took 15 years and $3 billion to complete the first human genome. Now it takes a few weeks, it costs less than $5 grand, you can do it yourself. Yeah. You guys have heard about, this specialized medicine, or what do they call it, customized medicine. You can pay and have your whole genome sequenced and find out everything that's wrong with you. I want to know. Okay, so recent advances in DNA sequencing have really revolutionized the field. So even small research groups, like me, can generate large amounts of sequence data quickly and at a fairly low cost. So take a look at this graph. So over here we have cost per genome over the years. Okay? I can sequence an entire bacterium for about $125. Or less now, depending on how many I send them, because they do it in bulk. Yeah?
What was it in 2008? There's a huge drop. Was there something that happened?
Next-gen sequencing, that's when it really hit, when Illumina technology and pyrus sequencing and these new techniques became available outside of just research labs, but you could pay somebody to do it for you. Yeah. It's a big, big change. All right. All right. So looking at what this has meant in terms of data in the field. So this is cumulative sequence genome. So this is numbers of genomes over time. Woo! It's still going. It's still going. I sequenced 13 just last year. All right. So let's do, as of August, I checked this in August, GenBank had, what is that? That's a billion, right? It's a lot. There's a lot of data out there. And that means we need people to be able to look at that data. You know? That's why I wanted to talk to you about this, because there's all this data sitting there. Here we go. Here's bacteria, because that's the ones I'm interested in, but showing you the numbers of whole, complete genomes, not bits and pieces, but complete genomes that have been sequenced over the years. And you can see that for bacteria, it's really going up quickly. But even so, we're at 15,000 for some of these others. Okay, so there's lots of data out there. Which leads me to wanting to tell you about in silico biological research. So what do you think that could be? What's in silico? Silicon. When we think about Silicon Valley, right? What do they do in Silicon Valley? They make computers, right? So this is computer-based research. Okay. Might sound kind of boring. I used to think that because I wanted to work on charismatic macrofauna, right? I wanted to work on sexy animals. But this is a really important field for the future. So we use this to mean that it's performed on a computer or via computer simulation. Anybody using computer simulation stuff in their classes? Right? Right, okay. So, and this is really important because it's thought to have the potential to speed the rate of discovery. Speed the rate of discovery, while reducing the need for expensive lab work and clinical trials. writing the programs, and you can come at it from that angle, all right? And some of the best bioinformatics projects are where the two work together. And we do some collaboration with some folks in the computer science department over at ODU to help us to develop new tools. And if you have a solid background in both, it's really helpful. So we need to tell our students that, and of course computer stuff, right? My 4-year-old can work my phone as well as I can. So we don't have to convince students to work on computers anymore, right? But maybe they don't know that they can do so much with it in terms of careers, like this. They know they can have lots of fun and become famous on YouTube, but, but to really make them think about it in terms of careers. All right. So, what can you do in this field? So once you're trained in the field, there are lots of different routes you can take. So you can be an individual contributor. That would kind of be me, mostly. So scientific labs in academia, commercial sector, you're hiring people that are trained in bioinformatics to support the research in the lab. Okay? And then we can go to core facilities. So for example, EBMS has a core lab where they can do sequencing. And so you have a person who runs that lab. And those jobs pay well, very stable, lots of opportunities to learn new techniques and grow in the field, so those are good jobs. Educators, so being able to teach bioinformatics, there are courses in bioinformatics at most major institutions these days. And then finally, and importantly, software developers. My updates for the software programs I use are not cheap. So that company is making money. And so if you know bioinformatics and can get into the software development side of things, you can do very well. All right? So those are some things in careers in bioinformatics. Other questions? Yeah?
I'm curious, what program do you upload the information and data collected? Is there a large group that you're working with to help them build a library?
Yes. So when I was showing the graphs that show all of the data as it's been accumulating, in the U.S. we mainly use what's called GenBank. And everybody deposits their stuff in GenBank. And GenBank is publicly available, and any of you could download sequences to play with your classes. Oh, yes. Yes, I know what you're talking about. I was thinking of a person, and I wasn't picturing it. Yes. No, I know what you're talking about. I haven't used it a lot, but I know that it's used often in classrooms because it's pretty student-friendly. Patrick is a program that you can use for phylogenetic tree building and things like that, right?
And problem solving.
Okay. Okay, so Patrick. Was there another question I thought? Yeah.
It might be too early to put on to ask this, but your work, you're cranking out the sequence. Do you have a question in mind, a goal, or you just getting info and then deciding what to do?
So you're at the crux of the question, which is, should I talk to you about my research, or should I show you an activity that you could do with this with middle schoolers?
Of an activity.
Activity.
But I guess it's
I don't know if I have enough time to do both. I don't think I have enough time to do both.
activity.
Activity. Okay. All right. So I do have slides about my research. Very quick version is I study a species of Streptococcus. So most of you know strep throat. So this is not particularly closely related to strep throat, and it's not a pathogen in humans. Well, actually, last month the first paper came out. Kid got stuck in the eye with a stick in India and got an infection in his eye with my bug. First human case ever reported. That's terrible. So it's not a human pathogen, it's a pathogen. It's primarily a pathogen of fish, here in the Chesapeake Bay even, which is where we discovered it, and cattle. Causes mastitis and dairy cow. So that's my, yeah. But we'll do our activity, yes? Okay.
One more question.
Yeah, sure.
On the careers, does it basically have to be master above to get a good job?
No. My students that leave my lab with bioinformatics skills have great opportunities getting into grad school. They're getting into Ph.D. programs with fellowships and making almost as much as I do. Seriously?
Wow.
Yeah, they are really in demand. If you have the skill Even if your GPA isn't the greatest, they want you. So you don't have to, you can even do it from a bachelor's degree.
That's what you asked.
Yes, you
You gotta get
can.
term.
Master's degree is better.
Yeah.
And actually, in what I hear is in terms of money versus time and
Right.
school balance,
Yeah.
master's is better. That's what I hear from the bioinformatics. Okay? Yeah. Yes. Let's do that on the break. We'll look it up together. Cool? Okay. All right. So, all right. I'm going to attempt this. I did a trial run with my students yesterday, so we'll see how we do. Yes. I need 18 volunteers. Back row, you get voluntold. So, I'm picking on you. All right, 18 volunteers. So she's passing out a piece of paper. While we're doing that, let me explain. Okay. So I have some resources for you here. So this will get uploaded so that you can get to these easily. And I decided to challenge myself to develop something. And I'm not a grade school teacher, right? So I'm sure that you will think of 20 things that could make this better. You may, go right ahead. But I wanted to challenge myself to come up with something for middle school. Because it seemed like this would be hard to do with middle schoolers, right? So I'm thinking about my kid who's in 7th grade, and what do I think he would do and enjoy? So that's why I developed this little thing I'm going to try. Okay, so these are here. All right. Tree of life. Got a picture in your head? Okay. Something like this. This is like a shorter one, but most of the time in a biology class you see a tree of life at some point. And students understand that it's like a family tree that shows who's related to who. Yes? So in this family tree, it's not just showing us who's related to who, but why we think that. Right? So we think that these guys down here are related to each other because they have hair, right? This isn't the perfect family tree. I spent a little while looking for it to try and find easy things because they're not going to know what an amniotic bag is, right? But 4 walking legs. Okay, we're good. Everybody here has 4 walking legs. So that's why they're related. Make sense? All right. So this isn't the only way, though, that we can decide if animals, I teach zoology, so I'm coming home. So animals are related to each other. There are other ways we can tell. So we can use genes to help us understand how animals are related. Wait a minute, what? How can I use genes? Okay, well let's start with what we know. What are genes made of? DNA, right? Your middle schoolers can do this, okay? Where are they found? In the nucleus, right? And what do they look like? So we have our answer right over here. Yes? This is what genes look like, because here's our DNA, and here are our A's and T's and G's and C's that make it up, okay? All right, so, all right, so maybe you will have talked about this, maybe not, but there's those A's and T's and G's and C's, and this is why they're important, because we use those from DNA to then make RNA, to then make proteins, okay? All right. So, how can we use genes to understand how animals are related? So here's our trial that we're going to do. All animals have a gene for the protein actin. They know what a protein is, we just talked about it, right? Protein actin. This is not scary. It's in all of our cells, and it's part of our muscles. Okay, we're good. I'm not scared, right? Since actin is protein, there must be DNA that encodes it, right? Because we just saw that, because it does DNA to RNA proteins. There has to be DNA that encodes actin, yes? Okay. So, for example, a butterfly might have this sequence. Okay, now the actual gene for actin is, I don't know, 1,000 base pairs long or whatever, but it's not important, right? So this is our little pretend sequence that we're going to work with. All right, so here's what it is for butterfly. So we can use this information to learn how other organisms are related to butterflies. Okay, so here we go. tree of life that's zoomed in to look at some of the bugs. And right here we have our butterfly. And here we have two empty boxes. And in those empty boxes should go either the fly or the bee. And so our goal is to figure out which one goes where. Which one is more closely related to the butterfly. Okay. So now, our volunteers, we have 3 groups. So I would like the butterflies there, the bees here, and the flies there. Butterflies, bees, flies.
What do you guys do? So then don't swallow them.
And then in addition, you'll
Show no
see
pest strip.
that your piece of paper has a number on it, yes?
Yep.
All right, and will you guys come so you can see the other groups?
Oh, yeah.
And you guys come down the side. Tom, will you guys come down the side so that you can, everybody can see each other? And then you put yourselves in order, 1, 2, 3, 4, 5, 6. What? We have a mutation. Is somebody in the wrong place? Good bee.
Mutation.
Here you are. Okay, put yourselves in order. Okay. If you're number 1 in your group, raise your hand. 1. Who's number 1? 1. Who's number 1? 1. Okay. So knowing where that starts, look at your sequences that you have here. Hold them up so everybody can see them. So looking at those sequences, which ones are more closely related to the other? All right, so we'll start with our butterfly, right? That's what we were starting with. So we have A, T, T, G, A, G. Okay? And then we're going to come over here. Here we have A, T, T, G, A, C. So that one doesn't match. Okay? So now we come over here and we have T, T, T. Okay, wait, so we already have a mismatch. T, 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 G, A, T. So which 2 are more closely related? Right. Okay. Now look at the tree. Where would you put, carefully, think before you answer, where would you put your fly? You guys are the bees, right? So we're saying the bees are more closely related to the butterfly. Where would you put the bee on the phylogenetic tree? Be careful. Look at it as a family tree. The bee and the butterfly. Bees and butterflies are more closely related. Okay, so I hear both answers. I hear the bee goes under the butterfly. I can hear the bee goes on top. Look at the branches. What do you think? Goes on top. Yeah. And remember, it's not how physically close together they are. Remember, this is
Follow the branch.
a family down here. This is like an outlier. Right? See it now? Okay. So, there. So we would put, oh, I forgot to put my little pictures. So the flies are here. I put my little fly here, and my little butterfly here, and my little bee here. So you did it. Yay!
So the bees are on the bottom? The bees on the bottom? No, bees on the top.
Oh, it should be the other way. It should be the other way. Yeah, it should be the other way. This is my first time doing this. Don't make that mistake. You don't have to read each So you guys can see how you can do this too, even at a really basic level. All right, Natasha, what do you think? 5 minutes? Okay. All right. And like I said, I just made that up.
Thank you.
And if I did what you guys do every day, I'm sure I could do a better job. This is your skill set, really. So I just wanted to give you a taste of how you can do this even at a middle school. Okay, I have 5 minutes, so I'll tell you just a little bit about research stuff. Okay, so this is my lab group. I'm very proud of them. So Jordan just started a Ph.D. program at Rice. Yeah, I'm really proud of him. I have another student at Baylor, and then these two ladies are my two seniors right now, and then I have two, a sophomore and a junior who are in my lab also, and actually a high school student also. So I already mentioned, this is the bug that I work on. It's called Streptococcus parauberus, and it's a gram-positive lactic acid bacterium. It causes infections in dairy cattle, so nursing moms may have mastitis. Mastitis has a really big impact on the dairy industry because milk from mastitic cows cannot be sold because it's all pus-filled. Yeah. Sorry, apologies. And it also causes infections in flatfish like flounder, or if you've heard of turbo in Europe, these are species of fish that are aquaculture. And so they cause these infections, and so in aquaculture they're not happy about this because they're losing their money when the fish are dying. To give you an idea, here's my bug down here, and then ones that you might know about. So strep throat, where's Pyogenes? This is right here, Pyogenes. All right, so it's not that closely related to ones that you know about for strep throat, but for a lot of people, there are other species here that they would know and go, okay, I know where I am. All right, we were the first ones to report this bug infecting finfish in North America, and we found it while we were studying striped bass. And we don't know publishable data, like solid data about prevalence. But what I do know is we found it never looked at. What's interesting about it, though, and why we got interested in studying it is it's doing all these bad things in flatfish, but in the striped bass here, they don't seem to care. Like when I look at tissues under the microscope, they don't look bad. The fish don't appear to be sick. So you should be thinking, well, then why are you studying it, right? Well, one of the reasons we want to study it is, why is that? Is that because the ones that live in the fish here are genetically different than the ones that cause infections in these other animals? That was where our question really started. And then, because if that's the case, then those strains that aren't so pathogenic, they can be used for vaccines, right? Because that's what we do with flu, right? When your kids are getting the flu mist, that's a live virus that's just not very pathogenic. So we can use it as a vaccine. The other reason is because we want to be able to track infections. And so I wanted to know, where did it come from? Did it come from dairy cattle on the Eastern Shore farms running off into the Chesapeake Bay? And so that was where our questions started. And so if that were true, if we look at the genes, we could say that the ones from cows look the same as the ones in the fish. Then we could say maybe that's where the infection came from. That's basic molecular epidemiology right there. And that didn't seem to be the case. What it looks like to us, this is my 5-minute version, what it looks like to us is that it's been here a while. That this bacterium has evolved to live in these fish and be relatively non-pathogenic as best we can tell. And they've made their own mutations that tell us that they've been together for a while and separate from everywhere else. Because when we do phylogenetic trees of these guys,
What kind of tissues are they found in?
we primarily are sampling from spleen, and we can just culture live stuff out of spleen. Crazy, right? So there's a phylogenetic tree showing our isolates from Chesapeake Bay in red. See how tightly they cluster? They're all very similar. They're not clonal, they're not identical, but they're very similar. They vary a little bit by river.
Yeah, my question. So, kind of out of the hat of the real scientific part. So, how a person likes each striped bass?
great question. That's a great question. If you're a person who likes to eat striped bass, there are so many parasites and nasty things in all of this fish that you better cook it.
Okay.
Irrespective of my bug, yeah, having done many, many dissections, as long as you cook it, you're fine.
Okay.
It's a very hard question. So we're comparing our bugs to those around the world. We have them from, 7 different countries, and Finland and New Zealand and the UK. So we're collecting them from all these places, and I really want to get the one from that kid's eye in India, so I can put it in my tree and see where it fits. Is it like really wacky different? It affected a person. Is it really wacky different? Or do we just not get sick because we're not sticking ourselves in the eye with it? So, okay.
So do all striped bass carry this?
No, not all. Not all. But we find it in at least some fish in every river. Yeah, Nanticoke, Potomac, Rappahannock. I forget who actually looked at New York. I think I still have samples in the freezer.
And they're not pathogenic to fish?
Computerbased. Right.
But they are to the cow? Is that the strain of the cow up top?
So that's, so we have on this tree, I have some cow strains that we collected from, this is the food science lab at Cornell. So all of these are from other places, from cattle, so that we could compare cattle strains and fish strains and see how different they are from one another. That's building the family tree. That's a really common thing to do, to build the family tree with pathogens. And then from that, try to understand how